Punnett square and monohybrid cross. The Punnett square is a graphical method named after its inventor, British geneticist Reginald Punnett. It is a widely used way of easily visualizing the variety of gamete combinations in breeding experiments. The Punnett square is a two-dimensional table that resembles a window pane. On this table, the gametes from one parent will be listed along the top edge, while gametes from the other parent will be listed vertically along the left-hand edge. These various combinations will give us an idea of the probable outcome of the cross. Let's create a Punnett square to see the possible outcomes of a monohybrid cross. Let's consider the color of a flower, which is controlled by a single gene. Each gene has two alternative forms which are called alleles. Red is the dominant color and white is recessive. A symbol will be designated for each allele in the gene pair. The dominant allele is represented by a capital letter R for the color red. The recessive allele is denoted by the corresponding lowercase letter, in this case lowercase r, for the color white. Because both parents produce the same genotype, capital R, lowercase r, it can produce only two types of gametes, either capital R or lowercase r. Now we'll write the gametes both horizontally and vertically so that we can determine the possible phenotype or external characteristics. For a homozygous dominant flower, the genotype would be capital R, a combination that will produce a red flower. If it is a heterozygous flower, the genotype would be capital R, lowercase r, and the flower it produces would also be red. However, if it is a homozygous recessive flower, the genotype would be lowercase r, and the resulting flower would be white. This phenotype will give us a ratio of 3:1, meaning there are three types of offspring which produce red flowers for every one offspring that produces white flowers. A Punnett square also illustrates genotype combinations. In this monohybrid cross, the homozygous dominant combination will be 1 out of 4. Meaning it will be 25% of the genotypes. The homozygous recessive will also be 1 out of 4, meaning 25%. The remaining heterozygous genotypes will be 2 out of 4, meaning 50%. The genotype ratio of a monohybrid cross is 1:2:1. Summary. The Punnett square is a graphical method used for visualizing the gamete combinations in breeding experiments. The phenotype ratio of a monohybrid cross is 3:1. The genotype ratio of a monohybrid cross is 1:2:1.
Have you ever wondered why your nose, eyes, ears and skin colors are the way they are? Do you know which of these traits have been passed down to you from your father and which from your mother? In 1866, an Austrian monk named Gregor Mendel published his findings about how traits are passed along from parents to offspring. These findings were based on his observations of patterns of inheritance in pea plants from one generation to the next. Mendel examined 7 distinct traits in pea plants. He found that there were 2 distinct forms for each trait, and that when one form was expressed, the other remained hidden. When he cross-pollinated plants that produce only yellow seeds with plants that produce only green seeds, Mendel found that the first generation offspring always had yellow seeds. The trait for green seeds seemed to have been suppressed in the first generation. In the next cycle, these first-generation plants were self-pollinated. In the second-generation offspring, for every 3 plants that bore yellow seeds, there was 1 plant that bore green seeds. This pattern of inheritance was observed for all the 7 traits Mendel had identified. Mendel also found that though a trait may not be expressed in an individual in one generation, it can still be passed on to the next generation. Mendel realized that certain factors were responsible for the passing on of these traits from parents to offspring, and that an offspring could receive only one form of each trait from each parent. Today, scientists know that traits are passed on from one generation to the next through genes which are carried by chromosomes. We also know that alleles are different forms of the same gene, and that each organism has 2 alleles for each trait. Based on Mendel's findings, for any particular trait, only 1 allele passes from each parent onto an offspring. Which allele is inherited from each parent is a matter of chance. When the 2 alleles for a trait are identical, they are homozygous for that trait. When the 2 alleles for a trait are distinct and different, they are heterozygous for that trait. In the offspring, the dominant trait will be expressed either when the offspring is homozygous dominant or when it is heterozygous. The recessive allele is not expressed in the presence of the dominant allele. A recessive trait only manifests when the alleles are homozygous recessive. The Punnett square is a simple graphical way of working out all the potential traits in an offspring based on the genetic contribution of each parent. Let's imagine that in a faraway universe, red eyes are completely dominant over grey eyes among the aliens. How will these traits be passed on to the offspring of a red-eyed and a grey-eyed alien? If one parent is homozygous dominant and the other is homozygous recessive, the first generation of offspring will receive 1 allele from each parent. In the presence of the dominant allele, the recessive allele will remain masked. Hence, all offspring will have red eyes. Now, in the next round, if a heterozygous dominant alien were to breed with another heterozygous dominant alien, in the second generation, 3 of the offspring will have red eyes and 1 will have grey eyes. Of these, 1 will be homozygous dominant, 2 will be heterozygous, and 1 will be homozygous recessive.
A species can be defined as a group of organisms that can interbreed and produce an offspring under natural conditions. Speciation is an evolutionary process that gives rise to new species. It occurs when variation is combined with geographic isolation of the species. An example of speciation can be seen with the snapping shrimp found in North and South America. As you can observe, snapping shrimps of Atlantic and Pacific Ocean are separated by the Isthmus of Panama. However, more than 3 million years ago, the Isthmus of Panama was being formed. The shrimp could move about freely through the Pacific and Atlantic Oceans and breed with each other. With the passage of time, the Isthmus of Panama formed a land bridge joining North and South America. The shrimps were separated on either side of the land bridge. Thus, they were said to be geographically isolated. The flow of genes among the shrimp was restricted to the group which they belonged. Over time, each group of shrimp changed genetically and adapted to new conditions. As part of a study conducted by the Smithsonian Tropical Research Institute, a male and female shrimp from different sides of the isthmus were put together. It was expected that they would court each other, but instead, the shrimp snapped at each other aggressively. Finally, the research team concluded that the shrimps on both sides of the isthmus had evolved into two distinct species.
What is accumulation of variation during reproduction? Have you ever seen an amoeba or an onion? They always look similar. Do you know why? This is because there is the only parent involved in asexual reproduction. So, you would find very minor differences between them. Variation occurs due to inaccuracies in DNA replication. In sexual reproduction, two parents are involved. So, the variations are very high. Variations are very important for evolution and form the basis of heredity. Let us understand the difference between heredity and evolution. Heredity is the passing of traits from the parents to offspring. This is how you get your features from your parents or grandparents. In similar way, evolution is a change in the heritable characteristics over successive generations. This is how humans have evolved over the years. After knowing heredity and evolution, let's understand inheritance. The transmission of genes from parents to offspring offers a common body structure, but differences in contrasting characters. Accumulation of variation happens when it is passed from generation to generation. Let's understand it using a simple example. In F generation, the cross-fertilization happens between the purple and the white flower, which results in a hybrid generation. When this fertilizes, it generates purple to white flowers in 3:1. This is how variations occur. During sexual reproduction, male and female parents take part in the process. So, the new offspring produced will have subtle differences. These offspring will give rise to another offsprings which will differ from each other. Examples of inherited variation through sexual reproduction are eye color, skin color, etc. Variation results in the formation of new species too. They adapt themselves to the particular environment and increases chances of survival. For instance, some rats in a population have a natural resistance to rat poison. Let us do a quick recap of the important points learnt in the video. What are the ways to classify organisms? Earth is the home to many species. According to scientists, there are far more species on Earth than what is discovered. Scientists have classified species into different groups based on characteristics. What do you mean by characteristics? Characteristics are the changes observed between different species rather than within species. In other words, details of appearance or behavior are observed of different organisms. They are classified into different groups based on these characteristics. This is known as classification. Most of the organisms share some basic characteristics. Classification can be done based on characteristics, similarities, dissimilarities, cells, etc. All organisms are made up of one or more cells. Organisms can be classified on whether the cell has a nucleus. Plant and animal cells have a nucleus inside the cytoplasm, whereas bacterial cells do not have a nucleus. Classification can also be done on basis whether organisms produce their own food through the process of photosynthesis. The skeleton that provides support, shape, and protection to the soft tissues are also used to make subgroups. It is not easy to study each and every species. Classifying organisms in groups helps to understand their life forms. It has taken years for scientists to study and classify living things. Most of the modern systems of classification are based on evolution of different types of organisms. What is evolution? Evolution refers to a slow, gradual, and continuous change in the heritable characteristics of living things. For instance, evolution of humans from apes. Although there has been changes in characteristics, but we have common ancestor. For instance, big family may have common grandparents. Hence, the classification and evolution are interlinked since evolution is development of new characteristics and the classification is done on basis of these new characteristics. Let us do a quick recap of the important points learnt in the video.
Puberty marks the beginning of a series of changes in the human body that lead to reproductive maturity. For a female, puberty is the beginning of her reproductive phase and can begin at around 10 to 12 years of age. At this time, when the ova or egg cells reach maturity, a single egg is released by one of the ovaries in a cycle of about 28 to 30 days. This is known as ovulation. The mature egg travels through the fallopian tube to the uterus. The wall of the uterus becomes thicker in order to receive the egg. If the egg is fertilized during this journey to the uterus and successfully implants, it results in pregnancy. If fertilization does not occur within 12 to 24 hours, the egg disintegrates and dissolves and is shed along with the lining of the uterus and its blood vessels. This shedding is called menstruation. The female reproductive phase begins from her first menstruation cycle and lasts until she reaches menopause. Conception or fertilization occurs in the fallopian tube. When a sperm cell meets and fuses with an ovum or egg cell, the resulting zygote continues the journey through the tube to the uterus. The uterine wall thickens to receive the zygote. As the zygote travels, it starts dividing and forms a mass of cells called blastocyst. The blastocyst gets implanted in the uterine wall and eventually becomes the embryo. The growing embryo continues to develop through the pregnancy until a baby is born. The gender of the baby is determined at the moment of fertilization. The instructions for sex determination are contained in chromosomes in the nucleus of our cells. These chromosomes are thread-like strands that carry genetic information. A human being normally has 46 chromosomes, or 23 pairs of chromosomes. Of these, 2 chromosomes, named X and Y, are called sex chromosomes. In humans, the female always has 2 X chromosomes, while the male has 1 X and 1 Y chromosome. The egg cells and sperm cells, or gametes, have only one set of 23 chromosomes. Hence, each gamete carries only one sex chromosome. So, while the female gamete will always have an X chromosome, the male gamete may have either an X chromosome or a Y chromosome. During fertilization, each gamete contributes one sex chromosome to the zygote. The egg cell with X chromosome may receive either an X chromosome or a Y chromosome from the sperm cell. If the sperm cell carries an X chromosome, the resulting zygote will be XX or female. If the sperm cell carries a Y chromosome, then the resulting zygote will be XY or male. Hence, the sex of the offspring is determined by the sex chromosome that is received from the sperm.
Why are acquired traits generally not inherited over generations? The baby girl has the same curly hair as her mother's. This is because the trait for curly hair type was passed from the mother to the child. All living organisms receive or inherit some traits from their parents, like the shape of the face, the shape of the nose, eye color, and hair color. These are called inherited traits. Inherited traits are encoded by genes present in the reproductive cells. So, inherited traits can be passed on to the next generation. Acquired traits develop or are learned during the organism's life, like learning to swim. Examples of acquired traits in humans are reading a book, playing a musical instrument, riding a bicycle, building body muscles. In a bird, learning to fly is an acquired trait. Acquired traits are not encoded by genes and so are not passed on to the next generation. So, only the genes or genetic changes in the reproductive tissues are inherited by the next generation. And these genetic changes are responsible for creating variations and supporting evolution. To understand variations and evolution, let us consider a population of 12 red beetles living in a green bush. Crows feed on these red beetles. In the first case, red beetles reproduce sexually and produce a green-colored variant. Crows spot red beetles in the green bush easily and eat them, while green beetles are camouflaged in the bush and remain safe. Eventually, all the red beetles are eaten up, and only green beetles survive and flourish. So, this is a case of genetic variation which aids the survival of green beetles. Such a natural selection leads to the evolution of red beetle into green beetle. Now, imagine that red beetles reproduce sexually and produce a blue-colored variant. Crows spot both the red and blue beetles in the green bush and eat them. The remaining red and blue beetles continue to reproduce and increase in number. But one day, an elephant stamps on this bush. By chance, all red beetles die, and only the blue beetles survive. These blue beetles are still being eaten by the crows, but the remaining blue beetles reproduce and flourish in this area. So, in this case, a random event changed the gene frequency of a small population living in a small area. Such an event is called genetic drift. Next, imagine a third case where the red beetles reproduce sexually to give all red-colored beetles. No variant is created. The bush suffers from a disease, causing it to shed the leaves that the beetles eat. Due to starvation, the size and weight of adult beetles decreases. There is no genetic change, and the change in size is an acquired trait which helps the organisms to survive. After a few years, when the plant disease is eliminated and there is plenty of food, the beetles grow in body size and weight. In this case, a change is caused in the body cells and not in the germ cells. This is why acquired traits cannot be passed on to the next generation. Thus, we learn that natural selection, genetic drift, and genetic mutation in gametes support the evolution of a species. Acquired traits, by contrast, cannot be passed on to the progeny and cannot contribute to evolution. Let us summarize what we have learned so far.
Sex determination. The sex of an organism is determined by specific chromosomes called sex chromosomes. In humans and other mammals, an XY sex determination system is followed. Every cell in humans, either male or female, has 23 pairs of chromosomes. Of them, 22 pairs are homologous and are called autosomes. The remaining 2 chromosomes are called sex chromosomes. In females, the sex chromosomal pair will be X and X, while in males, it will be X and Y. In humans, the X chromosome is not the same size and shape as the Y chromosome. In males, sperm cells are produced through meiosis with haploid or half the number of chromosomes, that is, 22 autosomes and 1 sex chromosome. Therefore, in a sperm, the sex chromosome can be either X or Y. Similarly, in females, the eggs have 22 autosomes and 1 sex chromosome, X. When a sperm with X chromosome fertilizes an egg with X chromosome, the offspring will be a female with paired X chromosomes. If a sperm with Y chromosome fertilizes an egg with X chromosome, the offspring will be a male with X and Y sex chromosomes. Summary. Sex of the offspring is determined by chromosomes called sex chromosomes. In humans, X and Y chromosomes determine the sex of the offspring. Females have X, and males have X and Y chromosomes.